Thank you very much for the opportunity to share some of the ongoing research that our lab is doing in this area. So really the core challenge that we often will face in the clinical setting is that when we do genetic testing on an individual, even sequencing their entire genome, we will identify millions of different non-coding variants. And it's really challenging to parse which one of these million or so non-coding variants are actually causative of the individual's condition in front of you in the clinic. With the advent of short read sequencing, we've been able to actually capture the contiguous sort of rate of which a genetic variation is accruing across the genome. But really the challenge has been this interpretation issue. One of the things that has been noted over the last several decades is that one way of actually improving upon the interpretation of these millions of genetic variants is by pairing genetic testing with functional data in the form of either transcript, chromatin, metabolomic, proteomic, you name it, these different type of functional data that can be really valuable in prioritizing which of these millions of variants is actually potentially causative of the individual's condition. And really the hypothesis and what I'm going to go over today is that a multiomic approach where you can obtain both this genomic and functional data directly from a patient can be used to help prioritize variants and also identify potential therapeutic avenues for individuals based on the mechanism by which you're actually able to resolve with patient samples in and of themselves. So before I get started, it's also really important to recognize that when we do genetic testing, we're taking the genome from this cellular context, which really contains a wealth of information in terms of the functional output of the genome. This includes proteins which are bound, which sort of turn on and off different segments of the genome, which can indicate areas that are functionally important. However, most genetic testing right now, what we do is we take all this lovely information and we essentially just strip the DNA away from it and just do genetic testing on the DNA itself without any sort of recognition of what this functional context was of the genome inside of the cell. And that's really the goal, is how can we actually move away from this genomic-centric based approach for testing and actually capture this functional information at the exact same time that we're actually undergoing the genomic testing in and of itself. Another important thing to consider is that humans were diploid. We don't just have one copy of the genome in every single cell. We have actually two copies of the genome. And differences in terms of not only the sequence, but also the function of these two copies is really critical for a lot of underlying rare diseases. In identifying functional spots along the genome that are distinct between these two copies can help identify and pinpoint specific genetic variants or epigenetic variants that may be causative of a given individual's disease. So how do we actually go about and map the function of the genome in terms of the epigenome? Historically, the main approach that we've used as a field has been essentially to use scissors. And what I mean by that is these are enzymes that will selectively cut the genome at spots along the genome of interest. This is in the form of Stergachis, TN5-based approaches, DNase-1-seq or aminase-based approaches as well. And these have been workhorses for the field of epigenomics and really have enabled us to have a really strong understanding of the catalog of gene regulatory elements that exist. However, there are some limitations with these approaches. And one of them is that it's really challenging to take these short reads that arise from these approaches and map them uniquely to each of the two copies of the genome that exist within each cell. And also it's really challenging to understand is how individual events are co-occurring on each individual chromatin fiber, which this co-occurrence can be really play a central role in how disease variants are actually causing disease. So how have we overcome this challenge? Really, instead of using scissors, we're using spray paint. And what I mean by that is that rather than cutting the genome at sites of interest, what we do is we take cells and we treat them with a non-specific adenine methyltransferase, which is essentially a spray paint that will methylate any adenine base that is not physically bound by a protein with this methylation mark. And since adenine methylation mark is really not endogenously present at any appreciable levels in humans and most multicellular organisms, if we see this methylation event, we can assume that that is a stencil of where exactly every protein was bound along that individual DNA molecule. We can then isolate these stenciled DNA molecules and sequence them using long-read sequencing approaches, which enable us to not only uncover the underlying genetic sequence of that long DNA molecule, but also a precise map of where proteins were bound along those molecules in the form of these M6A marked stencils. In addition to this, we also get CpG methylation information, which is read separately than the actual M6A methylation information. And both of these are natively obtained on sort of long-read sequencing technologies. We've now developed a suite of computational tools that take these stencils and turn them into functional inferences in terms of what exactly is functionally going on based on these stencils. And I'll walk you through a couple of these sort of tools over the next couple of slides, but really this is sort of trying to say is how exactly are individual regulatory elements actuated or positioned on a per molecule level, identifying individual transcription factor or other protein footprints, as well as using these primary stencils to directly uncover the 3D genome architecture of the sample of interest in a haplotype-aware manner. So just to walk you through sort of what this data looks like at its core, and this is an example of a locus, which is a known imprinted locus. And one of the benefits in every single, every single fiber on here is this gray line. And this is obviously cutting the fibers off just because you're only displaying around a 2 kb window. And these fibers on average are at least 15 to 20 kb in length. And what you're seeing here is every purple dash is a methylation mark. And so what you can appreciate is the predominant signal at this site you're appreciating is these sort of mononucleosome footprints, which are somewhat stochastically positioned on a per molecule basis. But you can see that there's a marked difference between these two haplotypes. And this is one of the benefits of grounding this technology in long-read sequencing is that it's relatively straightforward to phase our reads because the underlying DNA reads that we're getting out of this can readily overlap heterozygous variants, even if those heterozygous variants aren't contained within a regulatory element itself. We can now take these stencils or the raw data itself and use tools such as our Fiber-seq inferred regulatory element approach or FIRE pipeline, which now converts these into per molecule chromatin architectures. In this situation, I'm just coloring the individual inferred nucleosome footprints in gray, just standard run-of-the-mill intranucleosomal linker regions in this lavender color, and in red is individual per molecule actuated elements. And the benefit of this is twofold. One is it enables us to actually have a per molecule assessment of chromatin accessibility. And moreover, it enables us to actually pseudobulk our data based on haplotype. So I can now say, this is what haplotype, the paternal haplotype looks like in terms of chromatin accessibility versus this is what the maternal. And this really provides a strong power for looking at haplotype selective chromatin features. And I'll get into some of the importance as to where this comes into, so later on. So we've now done this on hundreds if not thousands of samples, and what we can find is that this approach is highly reproducible. This is just showing reproducibility in our FIRE scores across two replicates that actually diverged around 30% in their overall M6A stenciling rate, but nonetheless is able to correct for that. And furthermore, we've been able to develop this toolkit for both Fiber-seq library sequence using PacBio as well as ONT sequencing. This is just showing the same cell line sequence with both of those. But really one of the major benefits of this, in addition to the fact that this is haplotype resolved, is the fact that we get this per molecule assessment of chromatin accessibility. And what this enables us to do is to actually really for the first time ever have an intuitive metric of chromatin accessibility. And what I mean by that is historically, when you look at, say, a TAC-seq or DNase 1, you just are able to say this peak is taller than this one. But the actual unit measure of a peak doesn't really have any intuitive sense. It's sort of a reads per million or some sort of normalizing along those lines. Versus now I can say it's great. This peak is accessible on 95% of fibers versus this is accessible on, say, 50% versus this is accessible on 10%. And that really permits you to do not only more powerful statistical testing for looking at how regulatory elements are changed between samples or between haplotypes, but also gives a much more biological insights in terms of how the regulatory grammar is positioned at a per molecule level. Not only that, but what we found when actually analyzing this is that we're actually uncovering regulatory elements that previously were kind of challenging to identify with prior technologies. For example, this is a regulatory element right here, which is a well-known sort of CTCF binding element, which is a strong CTCF ChIP-seq signal. But if you were to look at the prior short read-based approaches, it had a relatively weak peak relative to these adjacent sites over here. And if you look at the Fiber-seq, though, this actually looks like a pretty strong Fiber-seq peak that's actually occupied on the majority of fibers. And what we found when looking through this data is that there appears to be actually a systemic underrepresentation in existing short read-based approaches in these short or small regulatory elements, like CTCF being one of the major ones in mammalian systems. And this is likely due to the actual kinetics of these scissors in terms of having actually cut multiple times within a smaller landing pad as opposed to a larger landing pad. And then also these probably short fragments being preferentially lost during library construction or a short read-based sequencing. And consistent with this, what we find is our Fiber-seq data is much more correlative with CTCF ChIP-seq signal than actually attack data is really emphasizing. We're getting a much more accurate representation of the chromatin epigenome with this single molecule-based approach. And just to further demonstrate that, what I'm showing you here is just peaks that we identify using Fiber-seq versus other short read-based technologies. And if you actually look at the enrichment of genome-wide association study variants, which GWAS variants should have been well described to be enriched in active regulatory elements, those peaks that are actually identified only with Fiber-seq and not with the ATAC-seq or DNase-1-seq are actually significantly more enriched in GWAS variants than those which are only identified in ATAC or DNase-1, really indicating that we're getting a cleaner profile in terms of the regulatory architecture and more accurate profile in terms of the functionally relevant regulatory architecture with Fiber-seq technology. Not only is that the case, but you also can look at more of the genome. You know, one of the benefits of being grounded in long read sequencing technology is that you can now map these reads to portions of the genome which have been completely off limits for short read-based technologies. This is, for example, the Notch2NL-B gene, which is a segmentally duplicated gene, which is present in 5 copies in most individuals, with greater than 99% sequence identity between each of those copies. Furthermore, this gene has been shown to be involved directly in sort of the human-specific expansion of our brain cortex, and so has been really sort of critical in terms of what makes us uniquely human. And what we can see now is doing Fiber-seq is that we can appreciate that there's a rich landscape of regulatory elements that are likely regulating this Notch2NL-B gene that are previously hidden due to mapping issues with short read-based approaches. And this is not unique. If you actually look across all of these segmentally duplicated regions of the human genome, which are some of the areas which are really make us unique as humans versus other primates, we're seeing a whole host of regulatory elements which have previously been hidden from prior-based sequencing approaches like ATAC-seq or DNase-1-seq, really emphasizing the ability of using a long-read-based chromatin approach for getting at more of the genome, which really we want to go after in terms of these core questions in human biology and evolution. So now bringing it back to sort of this question, which is how can we leverage this type of technology when it comes to actually the clinical setting? And really what the goal is that can we, rather than just doing genome, can we get this functional information at the same time, where we would then be able to sort of not just see and observe those variants, but actually functionally interpret them synchronously with the actual observation itself. For this, really the concept, which hopefully should be straightforward based on what I just presented in the last couple of minutes, is that we would take samples from an individual, do the Fiber-seq reaction directly on that primary tissue, then undergo sequencing with long-read sequencing to now get an accurate representation of the genome plus the CpG methylome and the chromatin epigenome. And for a first sort of test of this, we actually applied it to solve this condition, which is this non-goitrous congenital hypothyroidism. And specifically, this is a Mendelian condition which has been well described in the literature prior to this. And some of the genetic cause of this had previously been unknown, but actually there was around 12% of cases for which there was linkage to this locus on chromosome 15, but the underlying genetic basis or molecular basis of this was actually unknown at the time. Our collaborators using short-read genome sequencing on a number of families with this condition actually identified that there's a short tandem repeat in the contraction of the short tandem repeat from 4 copies of this TTTG to 3 copies was strongly associated with the presence of this condition, really indicating that this contraction is likely genetically causative of this condition in an autosomal dominant manner. However, one of the challenges that was faced by our collaborators at this time was that they have this variant, they have this condition, but they didn't really know how exactly this variant was mediating this condition. And the reason why is because this is a variant that was really located in the middle of nowhere between several genes, none of which were really associated with any thyroid phenotype. If you actually looked at the thyroid chromatin accessibility data, which was out there publicly available, it really doesn't look like a strong regulatory element or even a regulatory element at all. And moreover, this is actually located within a human or primate-specific ALU element, so it's in a repeat element, which makes it even more challenging in terms of mapping chromatin to this specific site. And so to better understand what's going on here, what we ended up actually doing is applying Fiber-seq to thyroid tissue from an individual with this condition that had been preserved in the freezer of one of our collaborators. And what we found is that when we applied Fiber-seq to this, and I'm going to separate out this by haplotype now, because just to remind you, this is a dominant condition. So one of the haplotypes should be the wild type sequence, and then the other haplotype is the one that actually should harbor this specific variant that we strongly suspect is causative of disease. And if you look at the wild type haplotype, it looks boring. There's really not that much going on. It looks very similar, our Fiber-seq data, to that of the existing DNase 1 data up here. However, if you actually look at the haplotype that contains this mutant, you can actually see that there's a lot of interesting stuff going on here. Specifically, you have actually the generation of multiple regulatory architectures right here surrounding this specific variant. And not only that, but you can appreciate down here, this particular promoter element is massively increased in terms of its chromatin actuation selectively on this haplotype. And we can show based on the phasing data that this is the only promoter within a 1 megabase region surrounding this particular variant that actually is selectively activated on this haplotype, really indicating that whatever is going on here is activating the promoter activity of this particular gene. But we can do better. We can actually zoom in now to this specific element to get a better understanding of what's actually going on. And this is where it benefits in terms of actually getting the single molecule and near nucleotide resolution of the Fiber-seq data. In this situation, if we look at the actual fibers from this mutant haplotype, we can appreciate there's a strong footprint that's directly and selectively occupying these fibers right over this site, and this corresponds to these adjacent FOXY1 binding elements. So what we're proposing with this is that actually you have this change in the spacing of these 2 pioneering factors, and that's actually now stabilizing their occupancy on this haplotype, which is then creating this cluster of regulatory elements to be selectively activated on this haplotype, which is then activating the promoter of that downstream gene. Furthermore, our collaborators were then able to do microRNA sequencing and actually show that what's actually happening is that downstream gene encodes 2 microRNAs, which are massively increased in their expression selectively in these individuals, and that actually causes a global thyroid dysregulation, which then causes the underlying disease. So one thing I want to emphasize with this case is that, these features of the mechanism, going all the way from the variant to understanding sort of the mechanistically how this is actually activating this microRNA gene promoter, were accomplished with a single experiment. It's a relatively straightforward experiment. This is, historically would be an entire PhD project, and this is now just one sort of experiment to actually accomplish that. In the latter parts, this part was actually just done using transcript sequencing, which really begged the question that we had in terms of the next case I'll present, which is, can we just combine Fiber-seq with long-read transcript sequencing to get better clarity and get this sort of all in one fell swoop? So with that, what we ended up doing was we, sort of layered on top of our chromatin data, sort of actually transcriptomic data, and for this, what we ended up doing was using a cDNA-based system, using the Connect system, and we now from individual samples are comparing both Fiber-seq plus long-read transcript data to get these functional readouts of the genome at the exact same time that you're actually getting the highly accurate genome sequence in and of itself. And just to sort of, visually illustrate sort of why this is a powerful approach and what sort of the benefits of this is, sort of what you would get at this individual locus if you had these haplotype resolved genetic information. So this is showing you the genetic variants on each of the 2 individual haplotypes at this given spot on the genome. But now once you actually layer on the CpG methylation information, the chromatin accessibility information, as well as the long-read transcript information, you can start to appreciate the rich differences that exist between these 2 haplotypes, not just in their sequence, but the functional output that they actually have within these cells, really highlighting the power of using this multiome approach, especially a long-read multiome approach, to resolving the mechanistic basis of how genetic variants are being interpreted within the cell. So one of the first cases that we ended up applying this to was actually this case, which was enrolled as part of our local undiagnosed diseases network here at the University of Washington and Seattle Children's Hospital. And specifically, this was a 9-month-old who had a whole host of different genetic issues, including a bilateral retinoblastomas, in addition to everything that I'm showing here before. There was no family history of these features, and genetic testing up to this point was really non-revealing in terms of the mechanism by which this specific condition was being mediated, including sequencing of the RB1 gene, as well as RNA sequencing of the RB1 gene on the clinical setting, which showed sort of this, maybe slightly lower RB1 expression. Prior karyotype of this individual actually did identify a balanced translocation involving chromosomes X and 13. And this was a question in terms of whether or not this translocation could be mediating her actual disease. Most individuals with balanced translocations actually do not have an underlying genetic issue as a result of that and are phenotypically normal. And so just by seeing a translocation doesn't actually mean that this is the cause of her condition. But also, since this is balanced, you have the normal haplotype as well, which is present in our cells, complicating the mapping with short reads if we were to do a standard short read based approach, because our reads would be sort of incorrectly potentially mapping across these 2 haplotypes. So really emphasizing this is sort of a case in where long reads can really play a beneficial role in terms of resolving this mechanism. So we ended up actually taking fibroblasts from this individual, performing long-read multiome sequencing on it. And with this, just using the genomic data alone, we're able to precisely sort of resolve the exact breakpoints of this translocation and demonstrate that actually these, along with the CpG methylation, the chromatin epigenomic full-length transcript data, as I'll show in the next couple of slides, that this ended up actually causing a quite complex phenotype at the molecular and mechanistic levels. And so at the genetic level, what we saw is that actually this particular variant disrupted the gene NBEA, and using the transcript sequencing, we were able to show that actually this results in a truncated NBEA protein and not actually nonsense-mediated decay. Furthermore, actually, this creates a fusion transcript between PDK3 and MAB21L1. This fusion transcript actually changes sort of the 3' UTR of this, and also ends up actually resulting in a enhancer that usually acts on MAB21L1, now accidentally acting on PDK3, resulting in the adoption of PDK3 of an enhancer for a different gene, thereby altering its tissue-based expression. Furthermore, this is a translocation that involves the X chromosome. We're able to show, based on the epigenetic data, that this results in actually spreading of exist-mediated silencing off of the sort of normal part of the X chromosome into actually this chromosome 13, and subsequently somatically silencing the RB1 gene locus in a small percentage of cells, but enough where actually that was enabled to initiate as the sort of initial hit in the formation of her bilateral retinoblastomas. And finally, we're able to show that actually this MAB21L1 gene ended up actually being silenced through a process called a transcriptional read-through silencing on one of the haplotypes. So it's a complex mechanism that we sort of resolved. This all sort of detailed in this paper from earlier this year. But really what I want to emphasize is that, if it were not for the functional data that we're getting from the epigenomics as well as transcriptomic aspects of this, we really would not have been able to resolve this mechanism at all, and may have been sort of stuck just at this top feature without understanding sort of the wealth of information that's caused sort of lower on here. So I really want to sort of emphasize this fact over the last couple of slides, that having this parity, not just of the genome, but actually this functional readout of the genome at the exact same time, can really play an important role in not only genetically, but also mechanistically pinpointing the cause of different rare diseases. And in the last couple of slides, I just want to sort of further just extend this, on the transcriptomic aspect of this, and just sort of remind people that when it comes to sort of long-read transcript sequencing, we're actually not getting a full capture of the transcriptome for multiple reasons. But one of which is that when it comes to Mendelian conditions, oftentimes a lot of the transcripts which we care most about are actually degraded by this concept of nonsense-mediated decay, because they contain either splice altering or premature truncating or sort of premature stop codons, you name it. And as a result, we're missing a lot of these functionally critical transcripts when we actually do standard steady state long read transcript sequencing. And so to resolve this concept in the next couple of slides, what we ended up actually doing was adding sort of an additional dimension to our long read multiome, where rather than just sort of taking cells and taking their RNA at steady state, we're also actually taking an aliquot where we treated them with this nonsense-mediated decay inhibitor, cyclohexamide, and that which is able to now stabilize these transcripts that would otherwise be decayed. So this is just showing you, just adding to this long-read multiome right here, and sort of the experimental paradigm that we had for this. For the next couple of slides, in terms of the cases that we applied this to, I also want to emphasize that, this wasn't a huge amount of sequencing we were doing. For this, we're doing around 30 to 60x coverage, Fiber-seq genome sequencing, around 5 million FLNC transcripts, across the steady state, as well as this nonsense-mediated decay stabilized approach. And we applied this actually as part of the Undiagnosed Diseases Network, as well as in collaboration with Boston Children's Hospital and the Manton Center for Orphan Diseases out there, across a host of individuals with really unknown condition, as well as several positive controls at the bottom. And these are cases in which truly they have had the sort of kitchen sink thrown at them from a diagnostic perspective, but have really been unable to identify what the underlying cause of the disease is, including both short as well as long read genomic sequencing, as well as short read RNA sequencing without any clear diagnosis up until this point, despite having sort of geneticists strongly suspecting that there could be an underlying genetic etiology. In the next couple of slides, I'm actually just going to be talking about our transcriptome analysis from this approach, but I really want to emphasize this is a tool which a graduate student in my lab created called IsoRanker, which really what the point of this was is, can we take the transcriptome data from these sort of long read multiome approaches and prioritize whether or not there's any genes that are being selectively disrupted in a genome-wide based approach. So this is actually doing a sort of function first as opposed to genetic first analysis. So if we were just to look at the functional output of the genome, can we identify functionally atypical or abnormal features that could then pinpoint an underlying genetic variant that may have been otherwise deprioritized or not prioritized at all in terms of analysis in a genomic first approach. We ended up developing this where it can actually test for multiple different aspects, including nonsense-mediated decay, expression outliers, as well as allelic imbalance. And then we tested this on a couple of sort of positive control samples. And importantly, when we did this, we're able to actually rank these within the top 10 genome-wide in terms of their importance, which means that if you're now a variant analyst, you would then be able to look at these specific genes and be like, aha, there's potentially an underlying genetic variant that actually could be driving this in this situation. These were the known cases where there was a variant that was actually driving these that we actually included. But more importantly, is the fact that when we actually took this and applied it to our cases, one of the cases really stood out in terms of something going on there. And specifically, actually, this was a case of an unsolved ataxia, or an individual with ataxia who actually already even had short-read RNA sequencing plus non-diagnostic genome prior to this particular test. And when we looked at the transcript data from this, what we saw is there's one gene in particular that really stood out, this HARS1 gene, which was ranked third genome-wide as being selectively subjected to nonsense-mediated decay, the first as being selectively loss of expression, and then the fourth in terms of its allelic imbalance. But not only that, there was actually, in addition to the loss of expression of HARS1, there appeared to be gain of expression of this fusion transcript between HARS1 and this gene DND1, really sort of raising the possibility that there could be a complex mechanism underlying this particular condition. So if we look at the actual individual reads, and this I'm separating out by haplotype, what you can appreciate is that there's more reads coming from HAP1 than HAP2. This is the steady state reads, and there may be some smidgen of sort of information coming out here that could be a fusion transcript between HARS1 and DND1. But once you now actually look at the ones after we've inhibited nonsense-mediated decay, what you can appreciate is that these reads that are actually the ones with this fusion gene with HARS1 and DND1 are now becoming much more prominent. And you're also appreciating that there's this transcript which skips this exon right here, which is also becoming much more prominent selectively on HAP2. And so this raises suspicion there could be a genetic variant either at this spot or somewhere over here that could be mediating this, and that suspicion is 100% correct. On this situation, on this second haplotype, we have a +5 spliced donor variant that's causing exon 9 skipping and subsequent nonsense-mediated decays, such that only 4% of the actual transcripts being produced on this haplotype are actually normally spliced at this site. And furthermore, on haplotype 1, there's a variant at this polyadenylation signal that is causing ablation of this polyadenylation signal on around 50-ish% of the transcripts. And on some transcripts, that polyadenylation signal is still being used appropriately right here, but some it sort of uses this polyadenylation signal that's a little bit further downstream. And then in some, it actually doesn't find any adjacent polyadenylation signals to use, so it just continues to transcribe through the downstream gene until it hits that gene's polyadenylation signal and then stops. But unfortunately, when this happens, these transcripts now have a stop codon right here, so they're all being degraded by nonsense-mediated decay, hence the fact that they're not actually being observed at appreciable levels in the steady-state transcript. Now, one critical aspect, and this is an important sort of thing to remember, is when you're doing these functional data or collecting this functional data directly on patient samples, you get functional information. And what I mean by that is based on this, we were able to say that on these two haplotypes, this individual is only producing around 28% of the normal transcripts for HARS1. And that number is really important when it comes to actually what we wanted to do next in terms of therapies. And what I mean by that is that individuals who have 50% activity of HARS1 or normal or higher don't have a human phenotype based on sort of individuals who are carriers for nonsense variants in this gene. It's assumed that individuals who were to have 0% HARS1 activity would not be living because this has actually been observed to be embryonic lethal in mice. And so it's, this is not sort of compatible with life. And so for this individual, he sort of lies somewhere in this particular range between 0 and 50%. And this is actually what we're sort of now observing is actually this multisystem ataxia syndrome, which is really matches perfectly with this individual and a couple of other cases that have been described in the literature with missense and nonsense variants in this gene that are biallelically in trans. But from a treatment or mechanistic perspective, it now sort of depends upon, well, what is HARS1? What is this gene? Why have I talked so much about it? So HARS1 is actually a gene that's important for taking histidine and loading it onto tRNA. And so this sort of charges tRNA with histidine. And in individuals with this condition, we can now say, well, it's just that you have less HARS1 present. And so you're just inefficiently loading histidine, and that inefficiency is likely what's driving their disease because they have fewer tRNAs loaded with this histidine. And this is important because actually the other RS1 genes have been well described where you can actually just overcome these defects through mass action. And what I mean by that is if you just give an individual more histidine, it will push this reaction forward and help compensate such that although only 28% of the transcripts are normal, you're still getting at least greater than 50% activity through this mass action concept. And this has actually been something which was recently sort of described in a clinical trial for individuals with HARS-related diseases, and something that our functional data was able to actually enable us to start this patient directly on supplementation 2 months ago now for this. So just to close out, really, I think what I'm trying to emphasize, and I hope you sort of get the point across with this, is that really the potential of long-read multiomics is that it enables us to not only just have this really accurate map of a patient's genetic variant information, which is really what's the power of long-read sequencing, but actually now start to understand what is the functional significance of these new or novel genetic variants that we're uncovering along this map. And I hope that you've seen sort of in the last couple of slides or throughout the talk sort of how this can be used not only for helping improve the diagnosis of individuals with rare diseases, but also actually getting at this concept of how we can actually start to think about this from a therapeutic perspective, thinking and interpreting a patient's own functional data in the context of their health. And with that, there's many people to thank for this project, or for the many projects sort of I did in here. This was, strongly collaborative work across multiple groups, both here at the University of Washington, as well as other groups here in the US and internationally, and also many of the funders who sort of enabled this project to be possible, including the NIH, the Burroughs Wellcome Fund, Pew, as well as Chan Zuckerberg Institute. For some of the funding for this. So with that, I'd be happy to take any questions.
Thank you, Andrew, for that presentation. So the audience has already submitted several questions, so let's get to them. So are there any differences between using PacBio or Oxford Nanopore for Fiber-seq?
Yeah, I'd say that there's a couple of differences to think about with this. One is actually just the resolving power of this. So this is a spray paint that we're methylating adenines. And if you sequence both strands of the DNA molecule, you end up actually getting resolution at both A and T bases because you'll get the A on the opposite strand. So with PacBio hi-fi sequencing, you end up actually getting a more precise measures of, say, transcription factor footprints because you have that, essentially doubling of the resolution because you can leverage both A and T strand bases when calling that. This is something that is possible with, let's say, a duplex mode of ONT where you're getting both strands of that. However, most individuals, when they're doing ONT sequencing, are usually just doing simplex mode of that. So you lose that resolution when sort of getting at that. But in terms of, say, nucleosome positioning, since nucleosomes are much larger, you typically don't see as much of a hit in terms of their ability to call nucleosomes with sort of the different technologies. So that approach is sort of less hindered by it. But really the transcription factor footprinting is one where you'll see the biggest difference between those technologies. And then the other sort of difference that we've seen, which is actually being improved upon thanks to sort of ongoing work at ONT, is traditionally when you actually did this stenciling approach, it actually had a significant impact on ONT's sequencing accuracy, although some of the newer sequencing callers or sequence callers that they have has sort of overcome some of these challenges. You still get a much sort of improved sequence accuracy with the PacBio-based approach, but the ONT is sort of coming up with that, which is good to see that progress on their front.
Fantastic. And how much optimization would need to be done using other cell types?
We've done a fair amount of cell types ourselves and also frozen and primary tissues. The optimization, I think, really comes, we've found, in sort of three flavors when you're actually sort of approaching a new sample. Most samples work out of the bat and without sort of any issues. Some, really what you need to make sure of is that you're not sort of completely over or under methylating the system. And this usually happens in situations where you just really inaccurately measured your nuclei count. I've seen this happen the most when, say, working with, say, flow sorted material, where it's like the flow sorter said that you had 1 million cells, but really you didn't have 1 million cells in that tube. For some reason, some are just lost, during the processing of it. where now the proportion of enzyme you're adding to the number of cells is quite divergent than what you had anticipated. The other challenge which I've seen is just actually around just the standard nuclei isolation itself. This is an enzyme which just needs nuclei to get in there. And so, if you don't actually permeabilize the cells appropriately, those fibers which are originating from cells that were not permeabilized will just not be sort of seeing the enzyme, and so therefore are not going to be as informative. And so really on that situation, just making sure that with a sort of new cell type you're working with, that your nuclei are appropriately permeabilized is, I think, an important step. And some that's pretty straightforward, like, for lymphoblastoid cells or PBMCs, that's actually been quite straightforward to work with. Some fibroblasts just may require a little bit more sort of aggressiveness in terms of making sure you're getting good nuclei out of that. Although we've had tons of phenomenal Fiber-seq data from fibroblasts, that's not an issue at all. And tissues, we've actually had really good success with as well in terms of using protocols. And then the other, I say, aspect is that, maybe the sample you had just didn't have actually chromatin on it. And what I mean by that is, working with, we've done this on some like old blood draws, where it's been sort of sitting around for a couple of days. And if you do that on old blood that's been sitting around for a couple of days, most of the DNA actually likely in that whole blood sample is just arising from neutrophils that have essentially apoptosed and sort of released their chromatin as something called chromatin nets. So you're actually not sequencing nuclear chromatin, you're sequencing this sort of extracellular chromatin nets, which for some applications may be useful, but for many applications is not necessarily what you want. So it's just sort of making sure that the sample you're working with still actually has chromatin situated on it. These are features that, we've processed, like I say, probably over 1,000 samples so far. We've had really good success with many samples, and there's actually good QC metrics that we've developed as part of this Fiber-seq QC package that help you measure whether or not there's any sort of these potential issues with your sample, even using very low pass sequencing, just to sort of get a sense of what's going on there.
And what about maybe fixed tissue? Is that possible, or is it very degraded at that point?
Yeah, I think it really depends on the degree of fixation that you do, whether or not it's a light fixation versus are you working with an FFPE sample. I would say that the major challenges you're going to face with the heavy fixation is just whether or not there's enough sort of intact DNA for it to actually be appropriately sequenced by the long-read sequencer. So like, are you actually getting that extra information or added information from that? From light fixation, I've seen that work through collaborators. We've historically just not done that just because we just haven't needed to or haven't had really the reason for doing it. And we've had really good success of identifying, transcription factor footprints and nucleosome footprints even without fixation. Like I showed you the case for the that thyroid sample where you could clearly see those footprints. Those were preserved there. That was not fixed material, but yet you can still see those really clean footprints that were critical for solving the mechanistic basis of their condition.
Great, and we wanted some clarification. Is this approach genome-wide and not targeted analysis?
Yeah. So Fiber-seq as core is a genome-wide based approach. We actually have a paper from either earlier this year or late last year, where we actually do have a targeted based version of Fiber-seq. And this is a methodology actually developed by Stephanie and the group who's on here. And this is sort of uses a Cas9 based system to selectively enrich for specific areas of interest along the genome and actually just sequence those areas after using the SAGE system for actually purifying out the DNA. We do have another additional technology called deaminase-assisted Fiber-seq, which does permit sort of the targeted enrichment of like specific amplicons along the genome and doing sort of that sequencing of those areas. And this is really a technology developed by Elliot as well as Easy in the lab, sort of to develop that deaminase-assisted Fiber-seq methodology.
Great. And what kind of special instruments are needed for this technique, or is it just the enzyme that's needed?
Just the enzyme. Like if you're working with tissues, Oftentimes for our tissues, we'll end up just doing a Down's homogenization, and sometimes we'll do a mortar and pestle sort of liquid nitrogen. But like there's many different ways of actually getting nuclei out of tissues outside of that. But for standard cells, it's, you take your cells, you isolate nuclei, which is typically either a IgE-PAL or digitonin-based approach or something along those lines. You take those nuclei, you treat them for 10 minutes at room temperature, with the enzyme. Then you undergo a standard sort of long-read DNA isolation. And at that point, you hand that off to the sequencing facility and ask them to give you some really beautiful data back.
That's great. And what is the minimal count of nuclei required?
Yeah, that's really a function of sort of what the sequencers can handle. And what I mean by that is that, this approach is at its core you're not doing any amplification. And so you need enough DNA at the end of the protocol such that you can actually generate a PacBio or ONT-based library for it. I would say comfortably with sort of the current, library prep protocols for those two, starting off with, say, 1 million mammalian cells is sort of will get you a comfortable amount. But oftentimes for some reactions where I'm really trying to sequence deeply, I may start off with a couple of million cells going into this. And this, from a tissue perspective, can be anywhere between like 10 to 50 milligrams of tissue is going to sort of get you sufficient enough on the human side. On the mouse side, you can typically get by with less tissue just because mouse just tend to have more nuclei per milligram, or at least in my hands when I've been working with that.
And can you talk a little bit about the bioinformatics pipeline for Fiber-seq? Like, is there specific example data sets available?
Yeah. So there's a couple of different bioinformatic tools we have. So one thing which is nice now is that the actual M6A methylation column for the PacBio instrument is actually done on the instrument itself. So that comes, like the BAM file you get off the sequencer has the M6A information already embedded in that. And the newer versions of Dorado for ONT also have M6A information that you can capture at the same time as CpG. So that means you have the stencils directly encoded in the BAM files that you're getting off the sequencer. From a processing for the FIRE pipeline, we actually have really good stuff that actually Mitchell Volger, who's really been leading this effort in the lab, has put together documentation, has a bunch of sort of test data sets as well to work with. And really the output of this pipeline is trying to get data in sort of standard formats that people are used to, i.e., genome-wide tracks of chromatin accessibility by haplotype. It calls peaks directly from the data. So like stuff that people are similar to are used to seeing for short-read-based approaches, but now having this added benefit of having haplotype resolution plus high accuracy with these approaches. In addition, we have a QC pipeline on our GitHub, and this is all sort of at this Fiber-seq GitHub here, where it enables you to sort of get these, really sort of straightforward QC metrics for this and out of this. So I would say that, this is a long read sequencing in terms of epigenomics is a emerging field. These are tools which we've, really been trying to pioneer and develop on, at the field. And, it's still not as developed as a field at the computational side, say as, short read based analysis of ChIP-seq. So I think there still is a little bit more of a hands-on approach in terms of long read analysis, but we have come so much in terms of where we are now versus where we were 2 years ago in terms of these pipelines and tools. Like they're much more robust. They're much more easier to use. They sort of, really enable this large scale analysis. And some of the emerging stuff that we're doing right now as part of the Human Pangenome Reference Consortium is actually enabling these tools not just to work with, say, HT38, but also sort of be working within a pangenomic context as well to sort of leverage some of the more emerging advances that are happening in the genome assembly realm.
And do you recommend long-read sequencing for FSHD testing? So this person is particularly talking about a D4Z4 repeat.
Yep. Yeah. So I think there's emerging data on that. I mean, FSHD, the current testing protocol, as I'm assuming this person is quite familiar with, is very complex. That involves, right now clinically we actually use a southern blot hybridization to actually do the testing. In addition to that, you actually need to know the genotype on which the testing was done. So this is sort of nasty flow chart that exists there. I hope that that will end up being replaced by long read sequencing. I don't have enough sort of knowledge about the specific sort of tools that have been developed, for just doing the genotype calling with the long read sequencing, but I do hope that that will end up being the case, just due to the clinical complexities that exist right now in terms of our current standard of practice for doing FSHD-based testing.
Perfect. And could endogenous M6A be a problem for the assay?
So really, I mean, in humans, endogenous M6A, if it exists, is at a level of, we measure this out to around 1 to 2 parts per million. That means that for every million adenine bases you sequenced, it's possible that one of those could have been endogenously methylated. So in humans, that level of endogenous methylation is really not at any degree which would be an issue. And we've similarly done this technology with Drosophila, we've done this in yeast, we've done this in mouse, primates, multiple different model organisms without any issue. If you were to say do this on a species that does have more endogenous adimethylation, so this would be C. reinhardtia or some of the other sort of species along those lines, which are sort of very specialized species, which have rates that are around like maybe 1%, which is around that's much higher. So this is like 10,000 parts per million, not 1 part per million. Then I would say that this may not be the right technology, but that is really rare organisms that people are using from a model system.
Perfect. Well, thank you for that discussion. We really appreciate your presence here today.
No worries. Thank you very much.